Hi everyone, myself Ritvi Waja and my partner Ayushi Patel, students of 4th year BMS, would be demonstrating to you a video tutorial on how to design primers for the desired sequence using primer blast in NCBI.
Primers are short stretch of DNA that target unique sequence and help identify unique part of genome, let's say a gene. Primer are usually 18 to 25 nucleotide long. They can be synthesized in a special lab and are used in a many different ways. For example, you can make multiple new copies of DNA from a template. You can introduce useful change to the template DNA where you can use them to analyze DNA by telling different versions of different copies. Primer selection and design prove to be one of the most important requirements in order to perform gene expression studies. The designed primers are used in polymerase chain reaction and DNA sequencing. The primer design tends to affect the efficiency and sensitivity of PCR. Primer design usually incorporate the use of two type of primers, forward and reverse primer. During the denaturation step, two independent strands of DNA are released wherein the forward primer bind to the template strand while the reverse primer bind to the complementary strand, amplifying both the strands. Rutvi will be showing how to design a primer, but it is important to know the design parameters first. So here it goes. Some of the important parameters that must be kept in mind while designing primers are listed below. So first is primer should contain 40 to 60% of GC content. Primer length should be in the range of 18 to 24 base pairs. Uh, Melting temperature should be more than 50 degrees Celsius and less than 65 degrees Celsius. The complementary percentage should be low from the reverse and forward primers in order to prevent dimerization. The number of repetitive nucleotides should be less in primers.
Now, since Ayushi has explained to you guys about the characteristics and what is primer designing, along with its importance, I'll take you to the hands-on step using NCBI Blast about how to demonstrate. I hope my screen is visible to you guys. So on your screen, you have to type NCBI. And after going to NCBI, we'll click on nucleotide because we are interested in the nucleotide sequences. So for instance, I'll type hexokinase and with that, I'll get my results. On my right hand side, you can see my cursor where you can see the organisms. So depending upon your area of interest, you can work on the organisms you are interested in. For now, we are just selecting the first sample because this is just a demonstration video. So once you click on the interested nucleotide sequence, you get this all. You have the FASTA sequence over here and the accession number over here. On my right hand side, we can see various different kinds of tools like blast, primers, highlight, sequence features, and etc. Pertaining to this video tutorial, I'll click on pick primers. So if I click on pick primers, I get the following screen which allows me to enter the variables for designing the primer. Over here, you can see that under accession number of FASTA sequence, it has directly copied from the datasets. Talking about the primer parameters, we are supposed to enter the primer parameters like for the reverse primers, forward primers, the number of things we want to involve like the PCR product size. Now, why this all is important? That is because in higher studies like the biotech studies and stuff like that, people want to have very precise locations of the primers because of the transcriptional or translational studies, and that's why we need to enter the forward primer and reverse primer. However, forward primer is just a DNA strand that attaches to the antisense strand of the DNA that runs in 3 to 5 direction. And reverse primers are DNA stretches that binds to the sense strand that is the positive strand of the DNA and that runs in 5 to 3 direction. This also helps us to select the exon and introns, which are also an important part of the nucleotide sequences. For your information, exons are termed as nucleic acid coding sequences which are present in mRNA, and introns are the non-coding sequences present in the DNA. which are removed by RNA splicing before translation. As Ayushi has mentioned, before designing your primer, we should ensure that the melting temperature should not be more than 50 degrees Celsius and less than 65 degrees Celsius. Also, the primer should have a GC content between 40 to 60%. So these are the two crucial parameters that we need to understand about. When you click on Get Primers, you'll get a window that would be loading your primer design. This might take 2 to 3 seconds more. That is because it would take time for designing the primer. So this is how your screen would look like, and it would take 2 to 3 seconds more to design the primer. So just to save time and not to make this video really long, I have already opened a file or the tab in which you can see that after clicking on Get Primers, what results you get. So this is the graphical view of primer parameters. So this is in general view, and if you're interested in particular regions like from 400 to 600 or from 600 to 800, you can click on it and you'll get the kind of the regions you are interested in. So it's like genes loading, and then you can see the genes of that particular sequence. Now you can see that it has designed 10 primers. Now it's up to us what primer we want to select based on our needs. So some wants primers to be concerned with only melting temperatures, so they'll see only TM portion. But some wants to be GS portion only, and they don't consider the TM portion. But however, a healthy primer is one which takes into consideration both of them. So over here, for example, you can see primer 1. They have given the forward primer length and the reverse primer length also. And they have also given the value for TMGC and self-complementary base pairs. So this is how you can see the 10 primers. And based on the interest of the study or the nature of the study, you can select the primer that you want. Before I end this video, I would like to brief you all about why is it crucial to design a primer. So a primer is needed to ensure the efficiency and specificity of the amplification reaction, resulting in a high yield of the desired amplicon. Now there are various other databases with which we can design the primers as well, such as RT Primer DB, Primer Bank, and some software that can help further on our Primer 3 and Primer Blast. Thank you for listening to our video.